topic I would like to talk about today is about clinical trials. Introduction to clinical trials. First, what are clinical trials? They are research studies that test a medical, surgical, or behavioral intervention in people. What do? They test new treatments or therapies to determine their safety, effectiveness, and side effects. They also provide essential data to ensure that new treatments are safe and beneficial before release in the public. Why are they important? They are vital for ensuring new treatments are safe and effective. They also drive medical advancements, leading to better healthcare and innovative treatments for various conditions. There are a total of four phases of clinical trials. The first phase is assessing initial safety and dosage. There are a small number of people in this phase, usually less than 100 people. The participants are people with specific diseases or conditions or healthy people. This phase is used to find the right dose of treatment to use in phase 2. They start with low dose and gradually increase the dose to see any side effects. In phase 2, they evaluate effectiveness and side effects. There are more participants, usually several hundred people. They begin to collect data on how well the treatments work in people who have disease or conditions. There are many ways to conduct a trial, but many trials use conduct group or placebo groups to compare with the other groups. They analyze specific gene and proteins that can indicate the treatment effect. The third phase is confirming efficacy compared to standard treatments. This phase has the largest amount of participants. It is conducted in multiple countries and includes thousands of people. They collect more information about how safe the drug is. They compare experimental with current standard to see if it works better. They also reassign participants into different treatment groups to get a better result. The last phase is ongoing study of long-term effects after approval. This phase is usually done after treatment is approved for use. It is to see how safe, how well it works in a diverse population. FDA monitors for public safety and potentially serious events. Biostatistics is used in clinical trial. First, what is biostatistics? It is an application of statistical techniques to scientific research in health-related fields, including medicine, biology, and public health. How is biostatistics used in clinical trials? It is used to ensure the statistical appropriateness of study design. It is also used to reduce bias. They use biostat to determine the optimal sample to detect a statistically significant effect. These models predict outcomes, measure treatment effects, and adjust for confounding variables. I will introduce some examples of clinical trials. First is the COVID-19 vaccine trial. It was an example of a good clinical trial. It was well structured that aimed to assess the safety and effectiveness of an mRNA-based COVID-19 vaccine. It was double-blinded, so neither participants nor researcher knew who received the vaccine or the placebo, which minimized bias. They calculated sample size, interim analysis, and clear criteria for endpoints. They received FDA emergency use authorization in 2020 and followed by full approval in 2021. The next is the initial Alzheimer's drug trial. It was a bad clinical trial because it had some flaws, such as it had small sample sizes and it lacked efficiency shown in interim analysis, such as some poor initial designs and potentially flawed assumptions. It was unblinded, leading to biases and unclear results. The analysis reported results with marginal statistical significance, which were not clear. Inconsistent trial design and poor biostatistical planning can lead to non-approval or significant skepticism. Thank you for listening.